Hello students, welcome back. Today, we are exploring the journey of T-cell development. Our journey begins in the bone marrow, where hematopoietic stem cells give rise to all blood cells, including the precursors of T-cells. These early T-cell progenitors leave the bone marrow and travel to the thymus for further development. The thymus, a specialized organ located in the chest, is the primary site for T-cell maturation. Here, T-cell progenitors, also known as thymocytes, undergo a series of developmental stages. These hematopoietic stem cells are giving rise to both myeloid and lymphoid progenitors. They are basically foundational cells in the bone marrow and have the remarkable ability to differentiate into various types of blood cells. Over here, these myeloid progenitors can give rise to red blood cells, platelets, monocytes, neutrophils, eosinophils, and basophils. And these lymphoid progenitors are differentiating into T cells, B cells, and natural killer cells. In the thymus, T cell progenitors enter the double negative 1 stage, where they lack CD4 and CD8 surface markers. Without Notch and GATA3 signals, these cells can become NK cells. When Notch and GATA3 signals are present, DN1 thymocytes progress to the double negative 2 stage. Here, they start rearranging their T-cell receptor genes. Some DN2 cells, while rearranging, can carry gamma and delta chain, others may proceed with alpha and beta chains. A successful TCR formation at the DN3 stage allows thymocytes to move to the double negative 4, DN4, stage, preparing for the next double positive stage. At this point, thymocytes express both CD4 and CD8 surface markers. They undergo positive selection to ensure their TCRs can appropriately recognize self-MHC molecules. Thymocytes that successfully recognize these MHC molecules receive survival signals and proceed to the next stage, where they encounter antigen-presenting cells for negative selection. This process eliminates thymocytes that strongly bind to self-antigens, preventing autoimmunity. Surviving thymocytes differentiate into single positive, SP, T cells, expressing either CD4 or CD8 markers. CD4 plus T cells become helper T cells, while CD8 plus T cells become cytotoxic T cells. So that's all for today's class. Thanks for watching. Don't forget to like and subscribe for more such videos.